Hi, my name is Lily Weitzel, and for my first research project, I did 3D printing of organs. What is 3D printing? Another word for 3D printing is bioprinting. Bioprinting is an advanced technology that is used, specially 3D printers, to create biotissues and organs that work like the ones in our body. The technology builds structure layer by layer using bioink made of cells and supportive materials. It holds great potential for creating replacement tissues for medical treatment and for studying diseases in the lab setting. The process bioprinting is a technology that carefully layers cells, biomaterials, and growth factors to build structure. These layers are arranged in a very precise way to copy how tissues and organs are naturally formed. The goal is to create tissues that not only look like real organs, but can also work like them. This technology is used to mimic the structure and function of real organs, which can help in the medical treatment and research. Next is application. Bioprinting can be used to produce organs that might one day be transplanted into patients who need them. It is also helpful for making tissue models that doctors and scientists can use for medical research. These models are useful for testing new medicines and studying how diseases affect the body. Next is components used, which are cells. These are living cells taken from the patient or donor. They are the main parts that make up tissues. Next is biomaterials. These are like the frame or support that helps shape the tissue and keep the cells in place. Then it is growth factors. These are a special substance that help the cells grow and work properly to make the tissues function. Some uses for bioprinting is organs transplants. Scientists are working to create organs like hearts, kidneys, and livers. These organs can be used to help patients who need transplants. Next is medical research. Bioprinting can make tissues that researchers use to study diseases and how they affect the body. These tissues are useful for testing new treatment. Next is drug testing. New medicines can be tested on printed tissues instead of using animals. This helps make drug testing faster, safer, and more ethical. Custom implants. Bioprinting can create implants designed to fit each patient perfectly. These implants are made to match the person's body exactly, which helps their function and be more comfortable. Some more are tissue repair. It can replace damaged tissue like skin, cartilage, and other body parts. Next is education. Doctors and students can use printed organs to practice surgery and learn about the body more than just testing it on real life people. Next is cancer studies. Scientists can print tumors to study cancer and test treatment in a controlled way. Reconstruction. Bioprinting can rebuild body parts like ears, noses to help patients after an injury or surgery. Some pros are solves organ shortages. Bioprinting can create organs which help reduce the need of donors. This can save the lives of many patients who are waiting for transplants. Custom fits for patients. The organs made with bioprinting can be designed to perfectly match the size and needs of each patient, making them a better fit. Less risk of rejection. When the patient's own cells are used to create the organ, the body is less likely to reject. This makes the transplant safer and more successful. Some more pros are it helps medical research. Like I said before, scientists can study using bioprinted tissue to study diseases and test treatment. This means we don't have to rely on animal testing anymore. The price could lower over time. When bioprinting becomes more advanced, it might cost less than a traditional organ transplant, making it more affordable for patients. Some cons are there still is a chance of body rejection. The immune system might not accept the new organ, causing problems for the patient. Next is restructure. The printed organ might not be strong enough to work as well as a natural one. And then incomplete function. Sometimes the organ might not be able to do everything a real organ can. And then, like I said, it could get cost less over time, but right now it is very costly. So only the wealthier patients right now can afford it just because it's so new, but maybe later on we will see it decrease. So high pricing is a con right now. And there is going to be errors in printing. Mistakes during the printing process can cause the organ to fail or not work properly. More cons are cell problems. The cells used in bioprinting may not survive or function properly once the organ is created, which can lead to failure. Next is an unknown long term effect. It is still not clear how well bioprinting organs will last over time. We don't know if they will work as well as natural organs in the long run. Ethical issues, these are concerns about how the technology is used, such as how cells are obtained or how it may affect the way we think about organ donations or transplants. Then we have limited materials. It can be difficult to find enough high quality cells in bioprinting to make or bioink to make bioprinting work well. The materials needed to just be right for the process and for it to be successful. Then we have regulations. The approval process for bioprinting organs may take a long time as a strict rule needs to be followed before they can be used in patients. Now we're going to go through the step by step. Number one is you're going to create a design. First, a digital model of the organ is made using an MRI or a CT scan to get the exact shape and details of the organ that need to be printed. Number two is prepare the bioink. Make living cells and mixed with a gel-like material to create the ink that will be used for printing. The bioink will allow the cells to grow and form the tissue. Number three is you're going to print layer by layer. A 3D printer is used to build the organ, one layer at a time. Each layer contains cells and biomaterials, and the printer arranges them precisely to form the shape of the organ. And then fourth is you're going to mature the organ. After printing the organ is placed in a controlled environment like a special incubator where it can grow and develop, allowing the cells to function properly and form a functional tissue. Next is test and use. Before the organ can be transplanted in a patient, it is carefully tested to ensure it works correctly, making sure it will perform just like a natural organ when placed in the body. And that is it for my first research project. Thank you.